Thank you. So, good morning, everyone. I'm quite happy to be here today. I will bring back DPP3 on some preclinical experiments. I'm sorry, but I promise it's the last time. I mean, at least from the Paris team. So, I mean, DPP3 is still a metallopeptidase. It's still present in the cells, and it can be also present in the circulation of acute diseased patients. To pick up just one cohort analysis we've done, this cohort of severely burned patients that are suffering from shock, where we saw that high levels of DPP3 were associated with circulatory failure, with systolic cardiac dysfunction, and with acute kidney injury. In the lab, I mean, using wild-type animals, we showed that the injection of DPP3 was able to decrease the cardiac function in those animals, as you can see here. Yeah, it works. On the other hand, using an antibody that was developed by Andreas Bergman in 144 company, protizumab, that we named PCZ. PCZ is a monoclonal antibody that will inhibit the enzymatic activity of circulating DPP3. And using this protizumab, we could actually reverse the cardiac dysfunction in a model of cardiac dysfunction induced by septic shock in rats. As it was said before, DPP3 is an enzyme, and one of the substrates of DPP3 in the circulation is angiotensin II. But there are very poor data, not poor, but very not so much data on DPP3, on the relation between DPP3 and blood pressure. There is this study that was published in 2016 by a Japanese team, the PANG study, which is a very nice study. The authors show that DPP3 infusion in hypertensive mice could decrease the blood pressure of those animals. What was interesting is that when they induce the hypertension using noradrenaline instead of angiotensin II, or when they use normotensive mice and they infuse DPP3, they had no effect on blood pressure. On the other side of the slide, there are the DPP3 knockout mice who showed increased levels of angiotensin II, for example, but those mice had normal blood pressure, which is a bit puzzling. So the aim of my presentation is to show you the data we have on how DPP3 can modulate blood pressure, the RAS, renin-angiotensin system in vivo, the blood pressure, and the kidney function. To start with the RAS, we injected wild-type mice with DPP3, protizumab, or PBS, and then we measured by mass spectrometry circulating RAS. As a control, you can see that the mice who had DPP3 have high DPP3, and the ones who received protizumab had lower DPP3 activity, which is normal. But when we measure angiotensin levels, we saw that DPP3 injection was associated with an excessive degradation of angiotensin II, but also of angiotensin IV, which was described being a substrate also of DPP3. On the other hand, protizumab injection was associated with an increased levels of angiotensin II, angiotensin 1-5, and angiotensin 1-7. Knowing now that DPP3 is able in vivo to degrade angiotensin II, we looked whether DPP3 can influence the blood pressure. So we took the same wild-type mice, not the same exactly, but we took wild-type mice, refinement, reduction in animal ethics stuff, but yeah, anyway. So wild-type mice injected with DPP3 or protizumab or PBS, but this time we measured blood pressure using a pressure transducer, but this was done invasively. and not by plethysmography. We did it also by plethysmography, and we couldn't see any change. And even invasively, we couldn't see any significant change of blood pressure in the mice that were injected with DPP3 or protzizumab. So we thought whether there is another system that can compensate for this degradation of angiotensins. We thought about catecholamines. It was quite easy thought, I must say. And it was really interesting, and we were lucky enough to see that DPP3 injection was associated with an increase of catecholamine release. On the other hand, when we inject protzizumab into wild-type animals, we could see a decrease of catecholamines release. Our hypothesis is that this catecholamines release post-DPP3 injection can compensate for the degradation of angiotensin II and then maintain a normal blood pressure. To verify this, we did another experiment, so we injected still DPP3 or protzizumab in wild-type mice, but this time we pre-injected the animals with labetalol, which is an alpha and beta adrenergic receptor blocker. And this time, when you inject DPP3 post-labetalol, you could see a decrease of blood pressure. And this confirms that the catecholamines release induced by DPP3 compensate for the degradation of angiotensin II and then the maintenance, and explain the maintenance of blood pressure in those animals. Our second question was, does DPP3 modulate the renal hemodynamics? This time, we didn't use the pressure transducer, but we used a flow meter. And what we observed is that when we inject DPP3, we saw an increase of the renal blood flow in the mice. And in opposition, when we inject PCZ, we saw a decrease of the renal blood flow. Blood pressure is stable, so this means that DPP3 decreases the renovascular resistance in those animals. To go furthermore in this, we looked whether DPP3, and to check if this effect of DPP3 on the renal blood flow is linked to angiotensin II degradation. We did this experiment. We checked the vasoreactivity of angiotensin II before and after DPP3 injection. So we took wild-type mice. We injected different range of angiotensin II doses, and then we injected DPP3, and then we re-injected exactly the same dose range angiotensin II. And we measured the mean arterial blood pressure and the renal blood flow before and after. And this is the variation that you are seeing. In gray is before, and in brown is after. And what we could see is that DPP3 administration is indeed associated with decreased systemic and renal vasoreactivity to angiotensin II. We then checked whether this effect of angiotensin II is AT1 receptor dependent. So we did the same experiment then for labetalol, but this time we pre-injected the animals with valsartan, which is an AT1 receptor blocker. And what we observed is that the effect of DPP3 on the renal blood flow post-valsartan injection was lower in the animals when compared to DPP3 injection without valsartan pre-injection, showing that at least partially AT1 receptor is involved in this effect induced by DPP3 on the renal blood flow. Another substrate of DPP3 substrates are enkephalins and endomorphins. So to check if they are involved in the effects we are seeing on the renovascular resistance, we pre-injected the animals with an opioid receptor antagonist, naloxone, and then we measured blood pressure and the renal blood flow. And what we observed is that this doesn't affect DPP3 effects on both, actually, which means that in this setting, enkephalins degradation by DPP3 are not involved in the renovascular effects induced by DPP3. It was already, shown several times in preclinical models and also associations in cohorts that in sepsis or in septic shock, the infusion of angiotensin II will decrease the renal blood flow, and this was associated with an improvement of the renal function. So to check whether this increase of the renal blood flow in our animals post-DPP3 injection may affect the renal function, we measured renal function biomarkers, creatinemia and urea, and we also measured glomerular filtration rates, and unfortunately we couldn't see any effect of DPP3 on the renal function in this setting again. So to conclude, we showed that DPP3 modulates the RAS in vivo. This was, the first time someone showed this. DPP3 decreases systemic and renal vasoreactivity to angiotensin II. This effect seems to be at least partially AT1 receptor dependent. DPP3 has no detectable short-term effect on the renal, on the kidney function. It's healthy animals. Half-life of DPP3 is 30 minutes. The renal blood flow effect we saw doesn't last more than 1 hour. So it might be that the setting is not very good to assess the renal function. And last, we saw that increased catecholamine release might explain why blood pressure is still normal despite decreased vasoconstrictive angiotensins. I would like to thank the team in Paris, especially Adrien Picot, who did all these experiments. I would like to thank also 14-4, especially Karine Bourgeois. And thank you.
Excellent mechanistic talk in 10 minutes, too. Very great job. Questions from the audience?
I think I killed everyone.
No. Well, then I have a question. So, the AT1 receptor seems to be fundamental in this pathway, and we heard talks about cardiogenic shock, and now these blood pressure findings. So, how do you suspect we should modulate the AT1 receptor in situations where we want to change, the impact of DPP3, or do we just not do that and look more for norepinephrine, et cetera, to sort of counterbalance the hypotension?
Thank you for your question. Something that Adrien also did during his PhD is we had this hypothesis that, there was a paper published saying that renin renin concentration and activity increase might reflect decreased angiotensin II concentration and might also reflect endothelial dysfunction leading to ACE decreased expression as well. And what we showed, because I showed only partially the RAS, but what we could see that is when you inject DPP3, so you decrease angiotensin II, you increase angiotensin I, so you increase the ratio angiotensin I to 2. We cannot measure angiotensins by mass spectrometry every day, but we have a very nice correlation with renin. So one can think of measuring renin, for example, to see whether this pathway is downregulated.
It makes sense. There's so many counter-regulatory functions of that pathway. We thought this was fundamental in COVID, right, with ACE2 binding, et cetera, and found that modulating that pathway didn't help. It may have caused harm. So there's so many feedback loops, and how do you actually modulate that to improve outcomes is quite challenging.
Yeah. And actually, the half-life of angiotensin II in the plasma is 10 seconds.
It's impossible to measure, yes. Yep. And even renin, right? With just changes in posture, your renin changes dramatically. So, questions?
I think that, of course, you thought about it. In what kind of disease it will be interesting to have DPP-3 as a drug?
You mean protzizumab or DPP-3? No, protzizumab. I mean, the clinical indication today, I will not talk in the, I mean, Karine can say it more better than me, but the clinical indication today is shock, septic shock, cardiogenic shock. But yeah, that's the first clinical indication for protzizumab.
Perfect.
I have a question because when you inject DPP-3 and when you see the catecholamine, the response is very quick.
Yeah.
And when you put AT1 blocker, it blocks. Do you think that it blocks at the level of the periphery, or it is a central blocking? Because in the hypothalamus, there's a lot of AT1, and it's a major regulator of
course.
Adrenal function.
Yeah. It's thank you, Nicola, for this question. We are part of the same lab. Yeah, it's a real option. Actually, one can think, when we inject DPP-3 and we measure the blood pressure, you couldn't see it actually on the graph I showed, but we had like 5 millimeter mercury decrease of the blood pressure, which might, then activate the splanchnic nerve and then the release of catecholamines. We can only answer this question by using renal denervation models, or by, using AT1 receptor and then blockers and then measuring catecholamines. But we cannot really exclude another mechanism of recapture of catecholamines, for example.
Thank you.
Thank you.
We don't have time. Thank you for all the speakers in this session.